One thing related to the duplication is the acceleration of evolution, accelerated rate of evolution. So showing this slide, there are a couple of cases to show after duplication, the lineage become much longer. So you already study, at least you practice on the computer program, knowing how to analyze or calculate KAKS. And also we can do comparison between different lineages. So here are the two cases show accelerate the evolution of new copy of new genes. And this is showing after gene duplication, one of the lineage accumulates extra amino acid changes along that lineage. And this is sort of like a very common feature of those new genes. And showing here, one of the case is the RNase, and I think both review paper mentioned about this case. And the second case is another gene discovered in Drosophila by Myron Long's group called Sphinx. So Sphinx, as you know from the Greek myths, is sort of like half of the lion, half of the human. So it's like a chimera of the new genes. So this is another thing we recognize. So from this new gene study, I want to illustrate not only the new gene, quite often after gene duplication, we also observe adaptive evolution or rapid evolution in certain lineages. Another thing discovered from all the richer genes, richer transposition now become a well-recognized mechanism in genome evolution. To generate new copies, new genetic materials in our genome. And also we can discover with the genome sequence, we can discover hundreds of richer genes. How we discover it? First, by homology. Second, as I say, the richer transposition usually use the material of the processed messenger RNA, the matured messenger RNA. So it lost the intron and sometimes still contain the polyadenation signals. So with all this feature, we can specifically, systematically study genome to discover all the possible richer genes or possible richer transposition events in a single genome. So one of the extended study is to understand the distribution or the movement of these copies. So this observation is based on first observed in Drosophila, but later in human and other organisms as well. So showing in this slide, Drosophila have 3 major chromosomes. X is the first chromosome, then second and third. But both second and third chromosome are both metacentric chromosomes, so have left arm and right arm. So this is just a little bit background about the chromosome, showing that this call it the x-axis. Then they try to first recognize all the richer genes, so they can scan through the genome, pick up all the possible richer genes based on the criteria I just described. Then they look at, okay, where is the genetic locus of or their parental copy? So this is all very new richer genes. So in the same genome, we can still recognize its ancestral copy. When we say its ancestral copy, means the copy generate the original richer transposition material that's matured messenger RNA. So by doing so, in the single genome, we can recognize both parental copy of the gene and the richer gene copy of the duplicates. So we have the direction. So you know which one is ancestral, which one is the derived. Usually in other type of duplication, with a single genome, you cannot really do that. So you have both copy exist functional in one single genome. You don't really know which one is the ancestral, which one is the derived copy. But you can compare to the close related species And by doing that, you just say, okay, what would be the most likely situation for the ancestral, the common ancestor? So that's the comparative genomics. But here, we can just use the gene structure and the sequence itself, decide which one is the parental copy, which one is the ancestral copy. Then right after they sort of find out all the research genes and their ancestral copy, they realize a pattern which is shown in this slide is the movement of the genes. So showing here on the y-axis is also the chromosome segments from X to 3R. So the slide showing the movement from one particular chromosome location to the other chromosome location. For example, if a dots showing here, it means original copy is on the 2L, then the end of it to the X chromosome, or the other way around. So showing on the table is more clear is the expectation or the observation, what they observe in the genome. And they look at if the ancestral copy is on the X chromosome and the reader gene moved to the autosome, that's the X to A. So that's the first one. And if the ancestral copy is on the autosome, then the reader gene is now on the X chromosome, it's the A to X. So these are the two major parts they are comparing to. Of course, there's majority of them are A to A, it's like originally in the autosome, then to the autosome. So the A to X is this part, and the A to A is on the part. And you can see obviously there's a lot of X's in the X to A pattern. The original copy is on the X chromosome, but now the reader gene copy is on the autosome. So this is like the axis compared to the expectation, because we can simply calculate the probability if this process is random. So because all the chromosome arms are pretty much equal in size, so you can just say, if this is a random process, what's the expectation? And compare to the expected number, what's our observation? It's the observation exceptional, means like a deviate from our expectation. So this is basically their observation, and it's really show the biased distribution of the reader genes. So this is like a study the reader transposition and showing the pattern like this. And of course they have the explanation, because X chromosome genes on the X chromosome for mammals and for Drosophila, they are the same. During gametogenesis, the sex-linked genes shut down much earlier than the rest of the genome. So think about if a gene expression is important, but it will eliminate early on in the gametogenesis, that could have like a strong effect for the fitness. So just by comparing the ideal biology, you can show the pattern is really a consequence of the natural selection. So talk about gene duplication, actually everybody would think about, oh, a gene duplicate and immediately have a second copy. But think of population. Remember in the beginning of our lectures, we talked about a mutation happened in the genome, and the mutation, once the mutation happened, the frequency of that new mutation is 1/2n. Duplication is the same thing. So duplication is a type of mutation. And when it happened to the genome, it means it happened only in one particular individual in that particular genome. So the frequency is just like other mutation, is very low. So this duplication has to go through the whole population process, become fixed in the population, then everybody has the second copy. Of course, there are like copies which is polymorphic in the population, just like in our genome, we have genes in different copy number. So not long ago, you see people talk about copy number variants in human population. So not all the genes duplicate will become fixed in the population. It's just like other mutations. So when we think about duplication, we have to think about like other process, like population process, and also selection drift. So all these evolutionary forces shaping the sort of like the final outcome of the duplicate. So this is just one of the slides I want to remind you think about in this direction.